The overall goal of this procedure is to identify involvement of novel components of ubiquitin and ubiquitin-like modifier systems in biological pathways, using the HIF-1 alpha-dependent hypoxia response as an exemplar. This is accomplished by first optimizing the response of the reporter cell line to the environmental stimulus, hypoxia, so they are suitable for screening. The second step of the procedure is to establish the high and low controls to give a suitable Z factor for screening. With the parameters established, the primary siRNA screen is performed in triplicate, after which hits are confirmed by secondary and tertiary deconvolution screens. Ultimately, the results can reveal new ubiquitin and ubiquitin-like system regulators of the biological pathway in question.
The main advantage of this technique over existing methods, like whole genome siRNA screening, is that the targeted screen is faster, easier to perform, less expensive, and reports only on the members of the biochemical pathway in which the investigator is most interested.
To characterize the hypoxia responsiveness of the U2OS HRE reporter cell line, have ready two 75 cm square flasks of cells in DMEM with 10% FBS. When the cells are at 80 to 90% confluence, aspirate the media from one flask and wash those cells twice with 10 ml of PBS. Trypsinize the cells and resuspend them in 8 ml of DMEM with 10% FBS. Make the cells homogeneous with a pipette.